Hi everyone, my name is Taylor Shirley and this is my presentation on stem cell culture. Stem cell cultures, also known as stem cell lines, are cells that are grown and maintained outside of where they naturally grow. These cells can originate from humans or animals and require specialized media for optimal growth. Stem cell culture ties into the cell culturing that we have previously discussed in Module 5. The cultures can be formed in either feeder-dependent or feeder-free systems. Feeder-dependent systems involve the stem cells co-culturing with feeder cells like fibroblasts. The feeder plates used in this method are shown in the top picture here, where the plate is conditioned with feeder cells, then plated with stem cells. Feeder-free systems, on the other hand, use extracellular matrix in the place of feeder cells. This matrix includes amino acids and other nutritional elements. The bottom picture shows the plates used in feeder-free systems, where the extracellular matrix is added into the medium when it is being made, then plated with the stem cells. The system used will depend on the stem cells being grown, with some require a feeder-dependent system and some requiring feeder-free systems. The controls used in stem cell cultures are positive control to show that the growth that would be expected from the cells, and a negative control that should not show any growth. One alternative to stem cell culturing is organoid culturing. Some features of stem cell culturing are that they are harvested from humans or animals. Cells harvested are limited to the amount of stem cells in the bone marrow being used. They can be maintained for over a year. Culturing is simpler than an organoid culturing, and they can be used in drug discovery. Whereas organoid culturing are derived from stem cells, replicate the complexity of organs, and can be limited by poor growth and patterning. On this slide, we have an image from a paper titled Advances and Challenges in Stem Cell Culture. In the figure, we see the human umbilical cord-derived CY3 labeled MSCs grown in 3D self-assembling scaffolds. Figure A shows a light micrograph of 2D cultured MSCs, with figures B through E showing confocal images of red fluorescent labeled self-assembling scaffolds in the absence and presence of collagen type 1. B and D both show the growth after 1 day under each condition, with C and E showing growth after 7 days for each condition. Here we have an image from a paper titled Embryonic Stem Cells. The figure shows the clusters of cells derived from murine embryonic stem cells, immunostained for surfactant protein C, a specific marker for type 2 pneumocytes. Here we have an image from a paper titled Culture Systems for Pluripotent Stem Cells. In the figure, both A and C show mouse and human embryonic stem cells on mouse embryonic fibroblast feeder layer, with B and D showing mouse and human embryonic bodies. Thank you for listening to my presentation. I hope you enjoyed, and if you have any questions, don't be afraid to reach out.